Thank you everybody for coming. Vance is currently on leave and hopefully having a great holiday, and Aimee was unfortunately not able to meet us today, but the end of this talk she has recorded a little bit of information. So what we thought we would do today was actually consolidate a few aspects regarding what we consider should be welfare considerations when thinking about developing arts and doing and implementing reproductive technologies for your captive amphibians. Basically, this talk is going to be centered around a paper that we put out earlier this year, which I've included the reference there at the bottom of the first slide. So yeah, I'll get started. Sorry. So sorry, I've got two screens going simultaneously, so I can try and coordinate this. So let me know if you have any issues, or let Kevin know, and feel free to interrupt me. So basically, we've talked about a lot already, is the fact that there's an ongoing amphibian crisis has been with us for decades now, and it doesn't seem to be ameliorating. And so, despite our collective concerted efforts as conservationists, mitigating the threats in the wild that are sort of affecting our declining populations has been really difficult. And really, if we're unfortunate, it's unfortunate to admit, but we're no closer to actually getting on top of any of these extinction crises. In fact, it seems like things may get worse going into the future due to obviously all of the climate issues that we're having, and habitat loss, etc. So whilst I'm basically preaching to the choir here, and I know that you guys all understand this, what part of this webinar series has been sort of bringing all of these issues to light and trying to convince the broader community how arts can help, but without making arts the final and only solution, and understanding that in order to have a successful captive breeding program, we really have to incorporate it as an accessory tool, and not hope that it's going to be the end all and be all of of any of the obstacles that we face in terms of reproduction. So obviously there are three main goals when it comes to the implementation of a captive breeding colony, and as such of the bringing of arts into it. And that's basically to prevent species extinction, to maintain genetically representative, survival assurance colonies of these species. I think that has to be our main focus. And then obviously, hopefully from there, provide future, provide offspring for re-release into habitats when we think that's a viable option, and it may not always be, which is part of the problem. Recently, as many of you may know, there's been a sort of a reevaluation of the Amphibian Conservation Action Plan, and compared to the other two versions that have been out, this is the first time that arts and biobanking is going to be featured as a chapter in itself, which is very exciting. And this map was just basically to give you a little bit of a preview to what we've been working on for this chapter, which is to tell you that we are sort of trying to establish where, when, and how these biobanking collections could start to actually help the amphibian community to salvage to store some of this genetic information, so that we can help sort of amplify the conservation strategies that are already in place. Kevin's recent publication on conservation needs assessment has shown that there's approximately, and correct me if I'm wrong here, Kevin, but I think from what I remember was about 2,300 species assessments have been carried out, and I know that there are still some areas of the world to finish doing, but what we did was, in collaboration with Kevin, was we took the quality this data and we actually mapped it out, and superimposed it on top of where for now anyway, Gina and I have been able to locate potential or existing biobanks that could be used around the world. But from the conservation needs assessments, as you can see, so far they've been concentrated in certain areas of the world. And if you look at where the biobanks are clustered, that doesn't necessarily, even though it's data deficient as to all of the species that need these assessments carried out, we can see that our biobanks are generally clustered as to be expected in certain areas of the world, and predominantly a lot of the time in areas which are considered of, kind of the more economically viable parts of the globe. But that doesn't always reflect where we have the majority of the species that need biobanking, at least so far, as per what Kevin's paper suggests needs to happen. So I just wanted to put it out there, that that's something that we need to be conscious of, especially for those of our colleagues of ours that are in areas where accessing these resources might be a bit harder. So we need to go into the future and sort of really assess how we're going to mitigate this as a community. So that's just a little bit, I realize it's a little bit of an aside, but I just wanted to put it out there, because what that boils down to is why all of us are actually interested in these webinars, is because we're interested in what arts can do for us. And sorry, I'm not very. And obviously, as we've reiterated, assisted breeding in captivity, assisted breeding, arts can help improve, hopefully, when natural breeding and husbandry approaches have been exhausted in terms of like what we think we've done, but we still can't get appropriate reproduction to happen, or we're not getting a good resilience of offspring produced. Then this is when obviously a good time where anybody should try to explore what arts could bring to that. It can hopefully improve on the genetic management of our species, and by either salvaging species from extinction or augmenting captive and wild populations. And we'll talk about that a little bit more going into this talk. And also, I think that at the end, what Aimee will discuss in the last part of the presentation, which she pre-recorded, will kind of give you an idea of how we can implement arts in terms of to increase genetic management strategies. And also, there's always the use of arts to improve reproductive health, not only overcoming the inhibition of natural breeding, but also to coordinate spawning and fertilization rates a little bit better, but also to avoid reproductive disorders such as egg retention, which can also affect not only the health of the founding and the adult population, but also have profound effects on the quality of offspring produced later. So again, before we go down the road of arts, just to reiterate that reproductive technologies and the techniques that we use for them need to be implemented along with good husbandry practices. I know I'm beating this like a dead horse, but I'm going to continue to do that so that it's so unbelievably irritating to you that you never forget it, because it will increase the likelihood that whatever protocols that you develop will be more effective. But not only that, we also need to understand going into these protocols and in trying to develop these, what we're trying to do with our ultimate goals, right? Because that's going to make an incredible difference it might actually affect the approach that we want to take when we're trying to mass produce animals. And I think this question came up in one of the last webinars. What is your ultimate goal with the implementation of your arts? If it's a conservation strategy, then we should look at, are we just starting an ex situ colony? Are we reintroducing offspring from that colony back into the wild? What are the other conservation strategies that are already underway, and how do these gel with our goals in terms of deliverables? And ultimately, if we're realistic, funding, right? Because we need to be able to understand the logistics and the financial side also of what we're doing with the implementation of arts and producing more individuals in a captive setting. So it is also important to understand that optimally the use of newer technologies can help us assess our species, that can help us assess, may not be available to everybody. And I go back to you know, the resource, the inequity of resources for conservation programs around the world. But basically, you know, this is a good time to really consider who to partner and collaborate with. In the case of this diagram, this is a very simplistic and broad roadmap to what I know Dr. Silla have worked on with respect to our own species and the programs that we've been involved with. And that's, you know, in collaboration with a number of people from different disciplines, we aim to combine our research areas in a more holistic approach to form a strategic management plan for our conservation programs, for amphibian conservation programs. And she will definitely talk about this more in with respect to one of her species that she works on, which is the northern corroboree frog, which is an iconic species here in Australia. And how we use and curate the genetic diversity within our captive populations is important. And knowing, for example, you know, the genetic structure of these populations going into from the beginning is obviously highly important, because then we can use arts like cryopreservation and hormone-induced spawning and IVF, or artificial fertilization, to actually really manage those genes, that genetic flow closely. And because this is the main, if we think that this is the main goal of or one of the main goals of a conservation, an amphibian conservation program, then, you know, using the most detailed methodology for determining the genetic structure of this species would be where I would start. That's not always possible, but that's, you know, in an ideal world, that's the first thing that I would do. And potentially start to look at replacing older methods of genetic curating, such as pedigree-based analysis, and maybe replace it for next generation sequencing, things like that. So we know from the beginning where our kinship, what kinship relationships we have within our populations, and that way, you know, really seek to preserve the greatest degree of adaptation within the species that we're trying to preserve. So again, we've talked about this a couple of times, so I'm not going to spend too much time on it, but, you know, required to initiate any reproductive events in captivity or in the wild, you know, we aim with our captive colonies to sort of recapitulate key environmental elements that will enhance or induce reproduction. And obviously this is not always easy, because amphibians display such an extreme mode of reproductive diversity, that setting this up is actually not always ideal in a captive setting. But knowing, at least beginning with knowing what kind of reproductive mode your species sort of fits into, gives you probably the most important foundation information from where to begin. And understanding, you know, a number of different reproductive traits, including whether, you know, their seasonality, their breeding structure, their age to sexual maturity, which may differ geographically and may also vary between populations that are in captivity versus in situ, climatic conditions, how those affect breeding. And if we break it down even more and we want to look at reproductive traits specifically, oviposition sites, egg and clutch characteristics, rate and duration of embryonic development, stage and size of hatching, even the presence and type of parental care will be important. All of these things would be, you know, ideally already known before we decide to start implementing any kind of assisted reproductive technologies. So I'm going to keep talking about this, but until now, what we have focused on is setting up the perfect environment required to initiate reproductive events. That's been sort of the focus of previous webinars so far. But to identify what key environmental cues work in synergy to increase not only the likelihood of breeding, but the overall quality of your offspring in captive populations. So when we talk about welfare, I'm not necessarily just talking about sort of a medical side of welfare, but when starting a captive colony, there are a few factors that I think welfare should also encompass, and we should talk about more broadly. And obviously, as I've just been saying, genetic structure of the population, because this not only gives you a snapshot of the species genetics and adaptive characteristics at the particular moment in time, but it also gives you a baseline from which to continually reference back to the wild populations if they're available. And that should be maintained over time. It's not just a question of bringing in animals or setting up a captive and then trying to maintain the diversity within that captive population without always going back to the wild and looking at how the flow of genetics is differing between the captive and the wild population. So genetic diversity of the captive population can and should continually be maintained and reciprocally transferred in and out of the population in the wild. And of course, that should be the main focus of thinking about implementation of ARTs, in my opinion. We should also understand or try to come to some sort of agreement if you have, if there's multiple stakeholders involved, on how genetically distinct your populations are, and if this is important, how important it is to maintain relatedness in the broader context of salvaging a species. Does inbreeding or outbreeding pose a threat to reproductive capability? How inbreeding or outbreeding affects maybe your chances of saving the species in general as well. And I'm not a geneticist, but that's I think a valid thing to think about when doing this. When it comes to founding populations, we should be combining this with knowing not only where the source populations arise, but the physical and chemical characteristics of that source environment. And the reason I'm saying that is because if you bring in a species, a group of individuals from a species that are already compromised, then you need to at least be aware that issue is there. So, for example, what is the quality of the areas where these animals are living or were living? Have we got data on things like pollution and water quality and general habitat quality? Because this is obviously going to affect across generations. It's going to affect the quality of the individual and the population that you're trying to save. What is their current resilience in those environments, and how does that compare to historical records? If you have that information, and I understand we don't always, then that's great. But I think even if you don't, it's probably worth having that in the back of your mind, or going back to those sites and looking at these parameters if you can, to try and at least understand what you're dealing with. So the other things that are becoming more researched recently are diet and abiotic factors, such as water quality. Do we truly understand what kind of food these animals consumed in the wild? Within CSCs, there is little literature. There's a little bit more literature coming out now addressing other aspects of husbandry on reproductive viability from the point of view of nutrition and water quality. For example, nutrition as an example is important most markedly in the case of females due to the high degree of energetic investment in yolk accumulation, which in turn is important to egg quality, which is then obviously tied into offspring survival. And very often, it's hard to know what the best nutrition for our captive animals is. And we're often sourcing those feeder insects from other farmed institutions. And so there are a lot of things that we really need to work on as a community to actually try and maximize our the quality of our captive colonies before we even start talking about NARDs. Mate choice is another one that I won't get too invested in because it's not really my area of expertise, but there is some evidence too that, and Aimee will talk about this in relation to the northern corroboree frog in a minute, and that's that even though it's a lesser reported aspect of reproduction, and certainly in amphibians that requires more research, the degree of mate choice in amphibians is very variable from the reports that I have looked at. And with respect to what effect it has on reproductive viability is obviously very species specific, and variable with respect to whether it's female or male mate choice that is more important in any particular species. So these are some of the other things that I've been thinking about in terms of welfare. Basic welfare considerations, obviously, when dealing with animals, and I don't think there'll be anybody in this webinar series who hasn't handled an amphibian, so you understand how to do this, I'm sure. When it comes to then administering things like injections, though, that's a little bit different, and so obviously when we're starting to talk about people using this on a more regular basis, then some of the things that we want to make sure people have access to is the proper training in order to be able to perform things like injections and understand how to grasp animals when doing things like egg stripping or sperm collection. Things to consider, very basic things to consider, obviously, are maybe getting injections ready before you give them to the animals to minimize handling time, and obviously, like I just said, restraint training. These things are things that will come up. I'm really going to just touch over them very briefly today, because as we go into the webinars 6 and 7 and onwards, these things are going to be broken down in a lot more detail, and the next webinar is actually going to show you a lot more about this. So Ruth and Dr. Silla and Dr. Phil Burns will actually kind of go into this more in detail. So obviously, one of the other things to consider is monitoring stress. And I think that a lot of us are already aware of sort of the basic physical characteristics that stress look like in our animals when we handle them. So that's one way to go about it. But I think obviously monitoring stress over a long period of time and looking at maybe chronic aspects of stress or not should also be implemented in terms of looking at hormone profiles. Again, this is something that needs to be acknowledged by the community, that not everybody's going to have access to, and so but nevertheless it does give us important information about what these animals are doing, and how they're responding to stress, to their environmental stresses, whether it be in the wild or in captivity. So again, just very briefly, because Ruth and Aimee will go into this, I think, in greater detail in the next webinar, but roughly there are 3 main types of, At present, there isn't a lot of research that's been undertaken to determine that stress response in amphibians after administration of repeated hormone applications. And so this is one of the things that I think the community needs to start actually really diving into, to sort of with respect to how the use of exogenous hormones affect animal welfare in the long run. But just very briefly, the route of hormone administration generally for those of us who do this on a regular basis has been mainly through injections, either intraperitoneal or subcutaneously. And now there is some more publications coming out to the use of topical hormone administration, which would definitely help in terms of not requiring maybe the training to inject animals. But again, Ruth and Aimee will talk more about that those are the three main ways that we go about administering hormones. Generally, you can do it intramuscularly, depends on the hormone that you're using, but that's more rare in case. So important considerations when using hormone injections, obviously, is, for example, that your protocols, as we've mentioned before, are going to be sex, not only species specific, but sex specific as well. And female aneurons are typically the more difficult sex to hormonally induce gamete release from. Female aneurons often require low dose primings prior to administration of an ovulatory dose to elicit that gamete release. And sometimes they may require multiple treatments or a combination of hormones at short intervals. So this is all part of protocol development that will be sort of discussed more in detail in the next webinar series. But that's an important thing to consider. Females have very different responses to the same hormones and their combinations thereof. It's also important to note that salamanders and aneurons also respond differently. So in salamanders, it is my understanding, at least, that salamander males are often quite difficult to induce with exogenous hormone administration. So depending again on the species, but salamanders seem to be an entirely different ballgame. And so I think Ruth will be talking more about that in the next webinar. And yeah, so whether they're continuous or discontinuous breeders is also important to know. My recommendations usually are to administer hormones in a manner that coordinates with natural breeding times, particularly if your animals are very seasonal, like most of the species that I have worked on are. There's some evidence to suggest induction out of season does work, and I think Vance spoke to that really early on in the second webinar. But it usually doesn't work out as well out of season, and so we recommend coordinating that with their natural reproductive times. There is not any published information to my knowledge about administering low priming doses early in the breeding season and sort of setting up like a cumulative effect. And that's probably because we don't usually have a lot of information about what the natural cycles of these animals are. And so actually designing protocols that could mimic that gradual increase in hormones is a lot harder when you actually don't know how that works or what kind of levels it occurs at. So one of the reasons, obviously, that I recommend anyway, trying to coordinate with breeding times is recommendations to administer hormones in a manner that coordinates with natural breeding times are also affixed to appropriate reproductive intervals between when gametes are deposited, because particularly in the case of females, there needs to be a time of restoration of body condition, right? In the case of both sexes, that's weight. We know that a lot of amphibians, particularly those that are from high altitudes and have that very sort of coming out of winter breed, and then don't even stop to eat and go straight into breeding, for example, they accumulate weight prior to their overwintering so that they can come out of that breed, and then they worry about eating and building that weight back up. So I think obviously looking at body condition is an important metric of understanding your recovery times. And understanding the natural reproductive intervals of these species, right? So again, if you have a species that typically breeds once a year really heavily and then stops, as opposed to maybe in a species that lives maybe further down in the tropics and has the capability to deposit multiple clutches in a year, then you're going to develop your hormone protocol strategies very differently for those animals. You also don't want to fall into, you want to avoid overstimulating your reproductive effort, particularly with females. because the cost of reproduction is so much higher in females than it is in males. And so, again, knowledge about the species' natural reproductive ecology is vital to be able to develop the best strategy going forward. But this isn't just important to the adults that you're breeding, it's also important to the overall fitness of the offspring that you're producing. So if females are in a poor condition, if males are overtaxed in terms of, you know, us spermiating them, then the quality of your offspring is probably also going to be affected. We definitely need more research in this area. This is more kind of based on anecdotal and, you know, life experience, but it needs to be definitely an area of research that we expand on, is actually looking at offspring quality under these circumstances. But certainly females, you know, poor body conditioning is probably also going to affect for things like, for example, yolk deposition. And if yolk deposition is compromised, then that will influence the quality of your offspring in a variety of ways. So as I said, I'm just in the next episode webinar for you guys in a couple of weeks is where Aimee and Ruth and Phil will really dive into the protocols for anurin and salamander exogenous hormone protocols. I just really wanted to sort of end off before I take you to Aimee's case study with just showing you some pictures of, you know, different ways in which we collect urine from males. There's a variety of ways that this can be done, and so it doesn't necessarily require somebody who's got a lot of experience. There are methods like what I call the gravity method, which is where we collect urine from frogs that are just sitting in damp containers. In this case, for example, a funnel, so the frog urinates and the urine falls into a receptacle in the bottom, and then you can pipette that off. So that's a pretty straightforward way to do it. Salamanders have, you know, people collect salamanders very differently than we usually collect anurins. So there's going to be more talk about this going forward, but that was just to get you going. Sorry. I'm just going to show you a couple of videos. This is from Ruth. She will talk about this more going forward, but essentially, you know, knowing how to handle these animals when you're trying to collect eggs, like she's doing in this video, this is what we call egg stripping, is going to be important and does require a little bit of training before we embark on this. In other instances, you can actually wait for your animals to, after administration of hormones, you can sort of, if you know the timing of when they start to lay, certainly sometimes you can rely on certain characteristics happening. This is the Southern Rocky Mountain boreal toad, and these females have these undulating contractions just as they start laying eggs. So that's pretty easy to pick up on if you know when to start looking. And certainly egg stripping from here would entail basically picking her up and just lightly holding on to those eggs that you see just popping out at the end and pulling them out gently. But yeah, there's different ways to start looking at how and when to collect these animals. So this is Aimee's portion of the talk, and as I said before, what she's going to do is actually give you the case study of the northern corroboree frog, and I think what it does is it really sums up a lot of the things that I've been talking about in terms of how and when to start considering using ARTs and how they can help increase the number of animals in captivity and the quality of your animals in captivity as well. So I hope you can hear this.
Thank you, Natalie, and hello, everybody. I am Dr. Aimee Silla, and I'll be spending the next few minutes discussing the application of reproductive technologies to a threatened amphibian. The northern corroboree frog, Pseudophrony pangilii, sister species to the imperiled southern corroboree frog, Pseudophrony corroboree. The northern corroboree frog is a small terrestrial toadlet. The species is restricted to alpine habitats above 850 metres altitude in the Brindabella and Fari ranges of New South Wales and the Australian capital.
Oh, sorry.
Thank you, Natalie, and hello, everybody. I am Dr. Aimee Silla, and I'll be spending the next few minutes discussing the application of reproductive technologies to a threatened amphibian. The northern corroboree frog, Pseudophrony pangilii, sister species to the imperiled southern corroboree frog, Pseudophrony corroboree. The northern corroboree frog is a small terrestrial toadlet. The species is restricted to alpine habitats above altitude in the Brindabella and Fari ranges of New South Wales and the Australian Capital Territory in southeastern Australia. Early observations document this species in great abundance, though the species has suffered severe and rapid population declines since the mid-1980s, primarily due to the amphibian chytrid fungus. The species is listed as critically endangered by state and federal governments and is listed as endangered by the IUCN. An insurance colony was first established for the northern corroboree frog in 2003 following collection of fertilised embryos from wild populations. The first viable clutch of eggs was laid in captivity in 2008, and this success was largely attributed to the realisation that the species requires appropriate temperature cycling, including a hibernation period, in order to come into breeding condition. If you're interested, you can read more about the requirements for natural reproduction of this species in captivity in the article that's listed at the bottom of this slide. Now, although the northern corroboree frog has been bred successfully in captivity for a number of years, captive populations display strong mating bias, with less than a third of available males contributing to mating success annually. Additionally, there are a small number of females each year that are clearly gravid, but they fail to breed. Our lab group recently completed a project determining the genetic mating system of this species using high-powered SNPs, and has shown that the natural mating system of this species also exhibits mating bias. You can read about that in Kelleher et al. 2021. However, it appears that mating biases are exacerbated in captive populations. Now, this is concerning because over time, such captive mating biases may lead to a loss of genetic variation and viability of the offspring that are generated for reintroduction. So myself and Philip Byrne, who co-head the Evolution and Assisted Reproduction Laboratory at the University of Wollongong, teamed up with Michael McFadden, supervisor of the Herpetofauna Department at Taronga Conservation Society Australia. And we decided to develop hormone-induced spawning protocols to assist with the genetic management of the captive breeding program for this species. Administration of a single injection per animal of GnRH analogue was used to induce spawning in isolated male-female pairs that were housed together in small aquaria. There was one male-female pair per enclosure, and we used 4 doses that were trialled: 0, 0.5, 1 and 2 micrograms per gram body weight of the animal of GnRH. Hormone therapy was very successful and increased the percentage of pairs spawning from 22 up to 100%. The optimal dose of 0.5 micrograms per gram GnRHA resulted in all pairs ovipositing a mean number of 31 eggs, which is above the average clutch size documented for this species in the field. Overall, in excess of 800 embryos were produced through hormone therapy over a 3-year study period. These offspring have all subsequently been released into natural sites in the northern Brindabella ranges. Importantly, the offspring released had greater genetic diversity because hormone therapy allowed a greater number of males and females to contribute their genes to the next generation, as opposed to a few stud males receiving all of the matings, in addition to a number of choosy females that failed to oviposit, which is what they would normally see. Now, given the enormous potential of reproductive technologies to assist with the genetic management of populations, why have amphibian breeding programs been so slow to adopt these protocols? Well, I think there are a number of reasons, and these might be touched on in one of the next webinar series when we discuss the hormone therapies used to induce spermiation and ovulation. But one of the factors that we believe comes into play is the need for user-friendly protocols which don't require specialist training in animal injections to apply hormones to our captive individuals. And so we have been developing methods for topical hormone application for our study species, the northern corroboree frog. GnRH is diluted in water instead of saline, and it is administered to the ventral abdominal surface of the frog. So it's applied just in a drop-wise manner. And this eliminates the need for injection. This approach has been successful at inducing spermiation in the roseate frog as well as a couple of other species, and it has proven successful at inducing spawning in the northern corroboree frog, which you can see here. My hope is that by developing such user-friendly approaches, it will result in more amphibian captive breeding programs adopting and benefiting from hormone therapies in the future. Now, while hormone-induced spawning has enormous potential to enhance propagation and can also allow for selective breeding of particular genotypes and ensure that all individuals within a captive colony are genetically represented in the offspring that are produced, to really maximize genetic diversity and adaptive potential, our ultimate goal is to develop and employ artificial fertilization techniques. Now, artificial fertilization protocols will be discussed in detail later in this webinar series. But earlier this year, experiments were successfully conducted for hormonally induced spermiation in isolated males, and sperm was biobanked as part of Taronga's cryobank. We were also successful at inducing egg release in females of the northern corroboree frog, as you can see in this video here. Unfortunately, I was not successful upon the first attempt to fertilize viable embryos, and so this is going to be the focus of research during the species' next breeding season, which is early in 2022. So with that, I'd just like to say thank you to my collaborators and funding bodies, and thank you all for listening. I'll now pass you back to Natalie.